Hello everyone. As you know that agriculture industry is a major part contributing to the world's economy, but many plants are destroyed due to pest or are destroyed by the disease. So how can we produce a plant which is free from the disease? And also as you know that the agriculture demand is increasing due to the increase in population. So now your aim is to produce a plant which is free from disease and plants of greater quality in a short span of time? The answer is plant cell and tissue culture. So, what is plant cell and tissue culture? The plant cell and tissue culture is a technique in which plant cell, seed, organ, or tissue is grown in laboratory under aseptic condition by the use of nutrient media. The characteristics of plant tissue culture include that environmental conditions are optimized, ability to give rise to callus, which is undifferentiated part, embryo or roots or shoots. Also, ability to grow as a single cell, that is in plant tissue culture, the cells can grow as the tissue can grow also as a single cell. Like in case of suspension cultures, the cells are dispersed. And also a thing to be remember here that in plant the cells are totipotent, that is, each cell can give rise to a whole organism, in our case, plant. The tissue culture has several requirements, which include that you have to be sure about the tissue that you are selecting because some tissue are better than the other. Some tissue culture are better than the others, such as seed culture, which is the best method for raising sterile seedlings. The media should contain all the requirements is that a cell need for it to be proliferate. This media can be liquid or solid like broth or agar. And the main thing in tissue culture is aseptic condition. You have to maintain aseptic environment and everything should be sterile, that is free from microorganism. So the output generated is free from microorganism or disease free plant is produced. Now media. So the media is should be sterile. And how it can be sterile? It can be sterile by autoclaving it at 250 degree Fahrenheit, which is 121 degrees Celsius at 15 psi for 15 minutes. The pH of media is usually is acidic, that is 5 to 5.7, and frequent subculturing can also be done to avoid the buildup of waste metabolites. The explant is the main thing in tissue culture because whole tissue culture depends upon explant. So explant is basically the startup for tissue culture. It is a small section or a small part which is taken from the plant, and this part can grow a whole plant. This explant is then sterilized by using surface sterilants or decontaminants. So to prevent it from any contamination, either bacterial or fungal contamination, and then it is placed in the culture vessel which contains agar and then it is left to grow. The X plant has certain characteristics. The This include that young portion of plants are more successful, which means that in case of X plant, the in case of X plant, the young portion are more successful. That is, if you are selecting an explant, you should be aware or you should be careful that the plant you are selecting is fresh and is young because it can then reproduce or proliferate more effectively and more efficiently than the other parts than the older portions. So that is, and also in explant the size and the source also depends, as I told you about the source earlier. So plant tissue culture is based on four stages. These four stages include establishment, multiplication, rooting, and acclimatization. Now let me tell you a bit about all these stages. In the first stage, which is establishment, the explant is selected and then it is disinfected. After which, in multiplication, the explant starts dividing and in multiplication and rooting stage, you maintain the hormone level as per the requirement. And in the last stage, which is acclimatization, the plant is exposed to the natural condition so that in the future it would cope up with the harsh environmental condition. So now let me tell you a bit about how the tissue culture is done. The first step is was the selection of X plant. So after you selected the X plant and the type of tissue, you rinse it with sterile water for 5 minutes or for 3 minutes for multiple times. And a more effective method is that you use surface sterilant or bleach, 20% bleach, and then disinfect it with it. The tissue culture technique is done in the laminar flow, and in laminar flow, everything should be sterile. So before using every equipment, you sterile it by placing it or by heating it under it become until it becomes red hot, and then you can use it. So by using this forceps, you place this explant into the agar, which is already present in the culture vessel. So now part goes to the brief explanation. So in the first stage, which is establishment. In establishment, the selection of explant is done. Depends upon the type of experiment and depend upon the requirements. So after the selection of so after the selection of explant, the disinfection is done. So for disinfection, you disinfected and all that is done. So after you selected the X plant, you picked it up with the forceps, which was sterile by heating, and then each of the X plant is placed in the media. Now, if you can see in the picture, here in the picture, the translucent part is the media and it is the explant. The explant is inserted by using forceps. And a thing to be remember here is that explant should remain intact and straight. The next stage is the multiplication stage. So in multiplication stage, the explant starts dividing and redividing, and due to which shoots are formed. The auxin and cytokinin ratio is maintained such that the cytokinin ratio is higher than the auxin because cytokinin favors the growth of shoots. So in multiplication, more shoots and branches are formed. Now let me show you the image. So in this image, as you can see, more shoots and uh, branches are formed. And in the next stage, which is dividing, as you can see here, that in this stage, multiple explants are present and in them more shoots are present than the roots. The next stage is the rooting. In the rooting, the auxin level is increased due to which more roots are formed. Auxin favors the growth of roots. Cytokinin is although present but in a limited quantity that it would only allow certain shoots to form. So as you can see here in this picture, more roots are formed than shoots. And the last stage is acclimatization. That is, now you have to acclimatize it or you have to introduce your plantlet to the environmental condition such that it starts adapting the environment, and then you place it into the ground or soil wherever you want. So here, as you can see in this picture, the plant is formed, and this plant is of great importance because it has no disease or it is free from contaminants or contamination, and this plant is produced is under aseptic condition. So this is the technique of plant tissue culture in which the plant produces free from the diseases and it is of greater quality, higher quality, more nutrition in case of fruits, and it is grown rapidly as compared to the plants which are grown naturally. So that's all. And if there are any question, please do ask in the comment section. Thank you.